नमस्कार, मी डॉक्टर मीनाक्षी राऊत, सचिव विभागीय मंडळ पुणे. महाराष्ट्र राज्य माध्यमिक व उच्च माध्यमिक शिक्षण मंडळ अर्थात आपल्या सर्वांचं HSC HSC बोर्ड पुणे विभागीय मंडळाच्या वतीने यावर्षी इयत्ता 12वी प्रात्यक्षिक परीक्षेसाठी शिक्षक, विद्यार्थी, पालक या सर्वच घटकांना मार्गदर्शन करण्यासाठी, माहिती देण्यासाठी व्हिडिओंची निर्मिती करण्यात आली आहे. यावर्षी फेब्रुवारी-मार्च 2026 इयत्ता 12वीची प्रात्यक्षिक परीक्षा 23 जानेवारी 2026 ते 9 फेब्रुवारी 2026 या दरम्यान होणार आहे. प्रात्यक्षिक परीक्षा घेण्यासाठी कनिष्ठ महाविद्यालयांनी, उच्च माध्यमिक विद्यालयांनी काय पूर्वतयारी करावी, प्रयोगांची मांडणी कशी करावी, प्रत्यक्ष प्रयोग कसे घ्यावेत, त्याचं मूल्यांकन कसं करावं, ॲक्टिव्हिटी, जर्नल इत्यादी सर्वच घटकांना गुणांची काय विभागणी आहे इत्यादीची सविस्तर माहिती या व्हिडिओमध्ये त्या विषयातील तज्ज्ञ अनुभवी शिक्षकांनी त्यांच्या प्रयोगशाळेतून लॅबमधून प्रत्यक्ष घेतलेली आहे आणि त्याच ठिकाणी सर्वांना मार्गदर्शन केलेलं आहे. या मार्गदर्शनाचा आपणा सर्वांना नक्कीच उपयोग होईल अशी अपेक्षा आहे. या उपक्रमासाठी महाराष्ट्र राज्य मंडळाचे अध्यक्ष आदरणीय डॉक्टर त्रिगुण कुलकर्णी सर यांचे मोलाचे मार्गदर्शन लाभले. त्याचबरोबर ही संकल्पना मांडणारे पुणे विभागीय मंडळाचे विभागीय अध्यक्ष माननीय औदुंबर उकिरडे सर यांचेही मार्गदर्शन लाभलं. राज्य मंडळाचे सचिव डॉक्टर दीपक माळी सर यांचेही बहुमोल मार्गदर्शन लाभले. त्याचबरोबर या व्हिडिओ निर्मितीमध्ये सर्व तज्ज्ञ मार्गदर्शकांनी अतिशय सविस्तर, सखोल अशी माहिती दिलेली आहे. या सर्वांचेही मी मनपूर्वक आभार मानते. त्याचबरोबर येणाऱ्या परीक्षेसाठी सर्व विद्यार्थी मित्रमैत्रिणींना मनपूर्वक हार्दिक शुभेच्छा. धन्यवाद.
HSC Board Practical Examination in Biology. Here we see the index number at right corner and the subject name code. The lab is well equipped and with all the instruments. Maximum 24 to 30 students have been taken for each batch. There are 3 questions for experiment section. First question is for major experiment. For major experiment, there are 3 options. First is potato osmoscope, second is peeling for study of plasmolysis, and third is structure and distribution of stomata. Both that question 4 marks has been given, 2 marks for preparation, 1 mark for observation and conclusion, and 1 mark for diagram. Second question is minor experiment. It is for 3 marks, 1 mark preparation, 1 mark observation, and 1 mark result or conclusion. Here is also 3 options are given. The first option is dissection of flower. Here is plant material used for flower dissection is hibiscus. The second is pollen germination. For pollen germination, 2 marks are for preparation and 1 mark for label diagram. And third option question is calculate population and density. In population density, the chart is given for students and they can calculate the density or frequency with the help of formula. The observation table consists of 2 marks and formula for 1 marks. Question 3 is minor experiments. There is 4 options. The first one is to perform or compare the given water sample for its pH and clarity. For clarity, 1 glass box is taken and the sample for water is passed from light and we can observe it. pH of water contains 2 marks for 2 samples and observation about clarity of 2 water sample contains 1. Second option for question number 3 is to test or compare soil sample for their pH and make note of their type. Here is also 2 soil samples given for pH and its type has been noted by student. Another question for question number 3 is make a temporary mount of given water sample and observe prepare report on presence of different phytoplanktons. Here 4 microscopic observed water samples contains 2 marks and inference content 1. Next part is part B. In part B, question number 4 consists of spotting. In this, 10 spots has been given to the students. Here identification contain half mark and explanation of relevant points contains half mark. Spot A. In this spot there are two options. First is wind pollination or insect pollination, that is salvia or maize, and second is controlled pollination, emasculation, bagging and tagging. Spot B also contains three options. First is rate of transpiration by four-leaf experiment, second imbibition of resins, and third is separation of plant pigments by paper chromatography. Spot C consists of morphological adaptation in aquatic and xeric habitats. We can mount or given any one aquatic or xeric plant in this spot. Spot D contains slide for TS of testis or TS of ovary or VS of blastula. Slide should be given under microscope and it is well settled that student can be easily seen it. Spot E contains syndrome from given karyotype. Here we can give the pictures and student can identify correct karyotype. Spot F contains identify any disease and comment on its control measures. Spot G contains meiosis slide. There is any one slide which is also shown in microscope. Student can observe it and explain it. Spot H contains adaptations of xeric and aquatic in animals. Spot I contains pairs of anthropozophil or model of eye, ear or brain. Spot J contains pedigree chart with respect inheritance of trait or Drosophila with eye color or wing type. Spotting, students should give 2 or 1 blank for describe their spots. Question number 5 is for project report, which is for 5 marks. Students should return and check this project with respective teachers. Question number 6 is viva or oral, which is taken by external or internal examination on question number 1 to question number 3. Question number 7 is practical notebook published by Balvarti. It is certified by respective teachers and external examiners should sign on this practical notebook. Here is now sample question paper has been shown. Part A contains major experiment, which is now we can see the sample question paper. Part A consists of major experiment for 4 marks. There are different questions related to question number 1. Second question is my minor experiment, which is contains 3 marks. Here is also 3 questions in second question. Minor experiment consists of question number 2 and question number 3. Question number 2 also contains 3 options and question number 3 it is also minor experiment for 3 marks. It is also consists of 3 to 4 options. Question number 4 is for spotting. There is A to J 10 spot. In each spot there are all respective spots given and teachers should select the different spot for different batch. Repeated spots cannot be seen in next batch, so we have two options for spotting and we can choose different for different batch. Part C consists of project report for 5 marks, vios for 2 marks and certified journal for 3 marks.